Imagine a world where two biological fathers could create a baby without a mother. Sound like science fiction? In 2023, a team of Japanese scientists took a giant leap toward that reality. For the first time ever, researchers created a living, healthy mammal, a baby mouse, from two male parents. And yes, she grew up, lived a normal mouse life, and even had babies of her own. So how is this possible? How do you make an egg cell from a male? Welcome to Mind and Marvel, where science, survival, and the superhuman collide. For over a century, one thing was considered biologically non-negotiable. Mammals need one egg and one sperm, from opposite sexes, to create life. But in March 2023, a research team led by Dr. Katsuhiko Hayashi of Osaka University broke that rule. Their question was audacious: can we turn male cells into female ones? It all started with a skin cell, from a male mouse. Just an ordinary cell. But scientists turned it into something extraordinary. They used induced pluripotent stem cell, iPSC technology. This process reprograms adult cells, like skin or blood, back into a versatile, stem cell-like state. iPSCs can become any kind of cell, including egg cells, under the right conditions. But there was a problem. These male stem cells had XY chromosomes, and you need XX chromosomes to make an egg. Here's where it gets wild. The scientists deleted the Y chromosome from these cells. Then, using a clever trick, they duplicated the X chromosome, creating XX cells from what used to be XY. This essentially feminized the male stem cells, making them genetically indistinguishable from natural female cells, and then, they guided those stem cells to develop into functional egg cells. These lab-grown eggs were fertilized with sperm, also from a male mouse. The embryos were then implanted into surrogate female mice. Out of 600 implanted embryos, only 7 live mice were born. One of them survived to adulthood. A healthy female mouse with 2 biological fathers. She grew up normally and even had babies of her own through conventional breeding. This breakthrough could reshape our understanding of reproduction. For people facing infertility, especially those who can't produce eggs, it offers a potential future path to biological parenthood. And for same-sex male couples, this might one day allow both parents to be genetic contributors to their children. But we're not there yet, not even close. The process had a very low success rate. Only 1% of embryos survived. We still don't fully understand the epigenetic risks, how reprogramming affects gene expression and long-term health. And while this works in mice, humans are far more complex. It could take decades to safely attempt something similar in people. There are also serious ethical questions. Should we do this, even if we can? Would it be used to heal or to enhance, to help families or to redesign them? Still, the implications are enormous. We're entering an era where the lines between biology and technology are blurring, where sex cells can be grown, edited, and created from scratch. This isn't just about 2 dads and a baby mouse. It's about challenging the core assumptions of life itself, and maybe, just maybe, reshaping what it means to create it. The future of reproduction is being written in petri dishes and DNA strands. Are we on the edge of a new kind of parenthood, or opening doors we don't yet understand? Let us know what you think in the comments below, and don't forget to like, subscribe, and turn on notifications.